afternoon, sir.
Afternoon, sir.
So let's start with the class. So I hope you remember last time we talked about monoclonal antibodies and their applications, right? And we discussed a lot of applications from diagnostics, from therapeutics, from research, right? So we discussed a lot of different applications of monoclonal antibodies with respect to you, it's used in therapeutics. We discussed a lot of different examples. I showed you a very long list, right? Although we did not discuss each and every therapeutic agent, right? Because at your level, it's not required to understand each and every uh, the drug or the monoclonal antibody, right? For treatment of various diseases, because then for that you'll have to understand the disease pathogenesis, you'll have to understand um, the process, this like the sequence or so many other things, right? So, but at least you should know few examples, right? You can remember few names and you can remember the mode of action of few of them, okay? So that suppose if a question comes on monoclonal antibodies or its application, then you can discuss that in detail, and obviously you can just go through the net, identify few examples and find the mechanism and other details from net, okay? And then you can always explain the whole thing in detail, okay? So in fact I think we discussed that last time also, that monoclonal antibodies are also being used for COVID treatment, right? So I will show you a small video on how again COVID and monoclonal antibodies, right? How they are or they can be used for uh, like therapeutics for COVID treatment. Okay, so let's first see this. We can have discussion after that, and then we will start with the new topic, which is your complement system. Okay, so let's first see this video.
Currently, there is no evidence-based targeted treatment for COVID-19, but the landscape for potential treatments is growing. One particular drug class, monoclonal antibodies, is generating some cautious optimism among researchers.
Many of us, I think, looking at the landscape would say, if there's a hope for something with a big effect size, I think there's evidence based on other circumstances, but nothing you can be sure of, is monoclonals derived from people who've survived COVID-19 ought to be our best hope here.
The first monoclonal antibody was approved by the FDA in 1986 for kidney transplant rejection. That antibody was based on murine hybridoma technology, which was invented a decade earlier. Murine meaning mice. First, researchers injected mice with an antigen, which is a substance that causes the body to make an immune response. Then, they isolated B cells from the spleen and fused those B cells with myelomas, or cancerous plasma cells. This creates hybridomas that produce antibodies like B cells, but replicate with the longevity of tumor cells. Hybridomas can be grown in culture, with each culture producing a single antibody targeting one specific antigen, hence monoclonal. And this allowed researchers to select the antibody with the right specificity, and then to mass produce it. Monoclonal antibody production has advanced a lot since the 80s, but it still follows those basic steps. You isolate antibodies against a particular antigen, select the best 1 to 3 candidates, and then produce a lot of them. And while some researchers still use mice to derive antibodies, the most popular method being deployed right now for the SARS-CoV-2 virus is to look right in the blood of recovered patients. an approach that was still aspirational less than a decade ago. Essentially, all research groups are focusing their efforts on antibodies that bind to the infamous spike, a well-exposed protein on coronaviruses that facilitates viral entry and that everyone loves to draw. The majority of these antibodies target the receptor binding domain of the spike, which docks into the ACE2 receptor on human cells. An antibody that binds somewhere along its 168 amino acids would have a good chance of neutralizing the virus. The blood of one recovered patient would most likely have multiple unique antibodies that bind to the spike and the receptor binding domain. However, not all of these would be equally potent, last long enough in blood, or fare well in the mass manufacturing process. So the companies developing monoclonals need to select the best candidates using a variety of in vitro and in vivo techniques. The good news is that more than a dozen companies are already past this stage. Some are going ahead with one carefully chosen antibody, while others are choosing a cocktail of antibodies, which might provide extra insurance that the virus will not escape or mutate away from the drug. So, which patients would be the best candidates to receive this type of drug? Well, a monoclonal that's safe and effective could definitely work to reduce progression of COVID-19, especially if given early. But an intriguing idea is to use monoclonals as a sort of temporary vaccine, especially in those at higher risk of exposure, like essential workers. And monoclonals that effectively prevent infection could be especially useful for vulnerable people who are at higher risk of developing severe or fatal disease, like nursing home residents.
We have many nursing homes that are suffering enormous trauma from this virus, and we might be able to go to a nursing home and give everybody in the nursing home right now a dose of cocktail of monoclonal antibodies, preventing infection. Vaccines might not work so well in older patients, and they take a while to work. So in the middle of an epidemic, a vaccine is less attractive than a monoclonal antibody.
Of course, a monoclonal antibody is not a replacement for a vaccine. Monoclonals would be effective for a few months, up to 3 or 4 if they have a naturally long half-life or have been modified to extend the half-life. Protection from vaccines is more long-lasting, at least a year and typically years, plural. But in the setting of a pandemic, monoclonals do have an important benefit over vaccines. Trials for monoclonals are much faster than they are for vaccines.
Vaccine studies that are planned are like 20, 30,000 patients. But monoclonal antibodies, there's a lot of reasons we can do much smaller trials and learn a lot really quickly, especially in places where the attack rate is very high. In the right clinical trial, we'll be able to tell pretty quickly, within months, whether there's a benefit or no benefit.
There are a lot of things about these drugs we don't know yet. For instance, what is their bioavailability in tissues affected by COVID-19? If the virus might mutate away from them, or what kind of adverse events they might cause. But the upcoming trial results, whether promising or not, will be crucial in deciding the course of this pandemic before a vaccine hopefully arrives. So look out for news over the coming months, since results from these trials will roll in as soon as August and continue through the fall of 2020. We've got a lot.
So, students, you just saw the basic principle about how monoclonal antibodies can be effective, right? And let me just draw this here to explain you. Is the whiteboard visible at your end?
Yes, sir. Yes, sir.
It's perfect. Now it's
Okay, okay. Okay, students, so okay, so suppose this is your host surface, okay, and this is your virus, right? and the virus will have a lot of different epitopes on its surface, right? Okay, and the host will have the receptor, okay, to which these epitopes will bind. Okay, so suppose you have the receptors. Okay, so when a virus enters our body, it attaches to one of the receptor. Okay, and when it binds here, then the virus gets internalized into the cell. Okay, so the first thing that need to happen is the interaction between the receptor and its protein on the virus. Okay, so what will happen now? This virus, it will go and suppose through this receptor, this epitope, it will go and bind to the receptor. Okay, but what we do is, so what monoclonal antibodies do is, so now we have developed monoclonal antibodies, and you can either develop it against the virus or against the receptor. The basic thing is that you need to uh, stop this interaction of this protein with the receptor, right? So what we can do is we can use monoclonal antibody and target these epitopes. Okay, now when the antibodies will bind here, and now if they go and try to bind to the receptor, they won't be able to bind it, right? Or you can develop a antibody against the receptor. So in that case, what will happen? Antibodies will go and bind to the receptor. Okay, so now your receptor is blocked, so this virus won't be able to bind to the receptor. Okay, so you can use both these approaches. Okay, and this will basically stop the interaction between the virus and the host. Okay, and therefore the virus will not be able to get inside the host or the host cell, right? So this is how these monoclonal antibodies are used for various diseases, okay, using this principal approach, right? Clear? Okay,
Yes, sir.
students? Okay. Okay. So now students, we have talked about antigen, we have talked about antibodies, we have talked about the different types of antibodies, we have talked about monoclonal antibodies, we have also talked about the applications of monoclonal antibody. Now, I think when I was talking about the different types of antibodies, I also discussed that some of these antibodies can activate the complement system, right? So today we will try to see what is this complement system and how this complement system works, right? And what is the importance of this complement system? What are the different types of the complement system? Okay, so we will talk about it. So let me share the slides. Okay, so is the slide visible?
Yes, sir.
Okay. So students, when we talk about complement system, so I think you can see image in the slide, right? But you, I'm sure at this point, you won't understand what it is, okay? So I will come back to this picture to make you understand what this is, what this ring is, right? So when we talk about complement system, so when initially it was identified, so Paul Ehrlich said that complement system is basically the activity of blood serum that completes the action of antibody. Okay, so something in the blood, okay, that completes the action of antibody. So you know antibody. What antibody does is it binds to the antigen, right? But that is not the ultimate goal of antibody, right? Immune system का काम सिर्फ एंटीजन को पकड़ना नहीं है, उसको एलिमिनेट भी करना है, उसको किल भी करना है, right? So if a pathogen has entered, it has to be captured by antibody and it has to be killed by the immune system, okay? So the activity of blood serum that completes the action of antibody. So antibody का काम bind करना है, okay? But this complement system will complete the action of antibody. That is, it will kill the pathogen. Okay, so just see here. So what happens in our body? We have a lot of complement proteins. Okay, so we have a lot of complement proteins, and you will realize in the next few classes that there are a lot of different types of complement proteins and how they are linked, right? So whenever a pathogen enters in our system, so you can see a pathogen has entered, and what happens? The complement system gets activated. The complement proteins, they get activated, and then they bind to the pathogen and ultimately kill the pathogen, right? So you can see this cartoon and how the complement systems proteins, they are very excited to see a pathogen because now they will get activated, and I will discuss how these complement proteins get activated. Okay, so they are there in our system, but they are not in an activated form. Okay, they are in our body, but they get activated only when required. So this is a kind of regulation in our system. Okay, because you know, if the complement system is always on, then it can have a lot of negative effects. So it has to be ready, but in a switch off way. Okay, and whenever required, they should get activated. Okay, when the immune system senses the presence of a pathogen, the complement system get activated. Okay, so there is a lot of signaling in that. A signal goes and messengers are produced, cytokines are produced. So a lot of things happen. Okay, so ultimately the whole idea of the complement system is to kill this pathogen. Okay, so let's see how this complement was discovered. So there were initial observations by Jules Baudet in 1890s. Okay, so he was the first one to talk about this complement system. He he observed that there is bacteriolytic activity. Okay, and that requires two different substances. Okay, one is specific antibacterial antibodies. Okay, so these initial observations were by Jules Baudet. Okay, and he said that there are two things. One is antibodies, right, which we have already seen, discussed in detail, and there is one more thing which is a heat sensitive component responsible for the lytic activity. Okay, so something apart from antibody which is responsible for the lysis of the pathogen. Okay, so at that time he just knew this. ठीक है, उसने अपने जितने भी एक्सपेरिमेंट्स किए, he was able to tell कि एक एंटीबॉडी रिक्वायर्ड है और सीरम के अंदर कोई हीट सेंसिटिव और भी कंपोनेंट है which is required for the lytic activity. इतना ही पता था. And then Paul Ehrlich, he also carried out similar experiments and named the substance complement. Okay, because it is complementing. Okay, and defining it as the activity of serum that completes the action of antibody. Okay, clear? And for this work on complement, Jules Bordet won the Nobel Prize for his work on complement mediated bacteriolysis. Okay, clear? So these are the two major scientists who were initially responsible for the discovery of the complement system. Initial observations were by Jules Bordet, and later on Paul Ehrlich also worked on this, and he termed this system as the complement system because he said that something apart from antibody is complementing the work of an antibody and leading to the lysis of the bacteria. Okay. Okay, so when we talk about this pathway, the complement system pathway, so there are three major types. Okay, the initial one which was discovered was the classical pathway. Okay, so this was the first pathway जिस पे Jules Bordet, Paul Ehrlich ने काम किया था. So that was the classical pathway. Okay. देन जब क्लासिकल पाथवे के बाद एक दूसरा पाथवे मिला, ओके, सो इट वाज टर्म्ड एज अल्टरनेटिव पाथवे. ओके, यू कैन अंडरस्टैंड कि एक तो क्लासिकल पाथवे और एक उसका एक अल्टरनेटिव पाथवे भी है. ओके, एंड देन लेटर ऑन अनदर पाथवे वाज आइडेंटिफाइड व्हिच इज कॉल्ड द मैनोज बाइंडिंग लेक्टिन पाथवे. ओके, और द MBL pathway. So at this moment, I'm sure you don't know what is MBL, that we are going to discuss later. Alternative pathway, again, we are going to discuss later. Today, we'll just start with the classical pathway. Okay. Now, in classical pathway, there is formation of antigen-antibody complex. Okay. So antigen पे antibody bind करेगी, and then the classical pathway will work. Okay, in alternative pathway there are some substances on the pathogen surface that will activate the alternative pathway. Okay, and in mannose binding lectin there is lectin binding to pathogen surface that activates the MBL pathway, right? So the three pathway, the initiation process is different. Okay, all the three pathways, difference कहाँ है? Initiation में. Okay, एक के लिए एंटीजन एंटीबॉडी कॉम्प्लेक्स बनना पड़ेगा, एक के लिए कोई पैथोजन सरफेस इज रिक्वायर्ड, स्पेसिफिक प्रोटीन्स आर रिक्वायर्ड, और एक के लिए लेक्टिन्स आर रिक्वायर्ड, राइट? एक बार इनिशिएट हो जाते हैं, सो आफ्टर फ्यू स्टेप्स all merge at a common point, and ultimately it leads to complement activation, and then complement can do a lot of things, right? Let's see what it does. One of the most important thing is lysis, right? और Paul Ehrlich और Jules Bordet ने भी यही कहा था कि apart from antibody there is something else which is required for the lysis of bacteria. Okay, lysis होगी तो bacteria kill होगा, right? Do you understand this? So you can see the complement system will ultimately lyse the target cell and kill this cell, right? Another thing is opsonization, right? Another function of the complement system is opsonization, and I think I have discussed this term with you. So students, does anyone remember what is opsonization? Does anyone remember what is opsonization?
Sir, it's basically a way to make any kind of invading pathogen. It makes it easier for the phagocyte to digest.
Yes, yes, right. So basically, it helps in the process of phagocytosis. So the complement proteins or the complement system proteins, right? They help or they make the bacteria more vulnerable for phagocytosis. Okay, and you can see here in this picture that these complement proteins, these red protein, they will bind to the bacteria. Okay, and that will help in the uh, uptake of or the phagocytosis of bacteria, right? And obviously, once this bacteria is inside the phagocyte, it will be killed by the phagocyte, right? Another important role of complement system is the activation of inflammatory response. So you know what is inflammation, right? There are 5 different features of inflammation. So we will also discuss that in some other class. But complement system also activate the inflammatory response. Okay, and another very important role is clearance of the immune complexes, right? हमारी बॉडी के अंदर when some pathogen enters, okay, so the antigen is circulating, right? So in this antibodies will bind. Okay, now this complex, right, it cannot just keep roaming in our system. जब इसका काम खत्म हो गया, it has to be cleared off. Okay, so again this complement system, it helps in the removal of the immune complexes or clearance of the immune complexes from the system. So now I think with this slide, it shows you that there are 3 different pathways of complement system. Okay, and there are various functions. So 4 of them, the main one are shown here. Okay, one of them is the lysis or the killing of the target cell. Okay, which is the most important thing, because ultimate aim of the immune system is to eliminate any pathogen. Okay, then opsonization, again, it's basically killing of the pathogen. Okay, the mechanism is different, but the complement system is playing a role here because it releases the complement proteins which go and bind to the surface of bacteria, and then the bacteria is taken up by the phagocytic cells. Now how this happens we will discuss later, because it has to be the role of some receptor, right? So that is second important role. Third, as I said, is the activation of inflammatory response. We will again discuss inflammation in detail in some other class, and then clearance of the immune complex. Clear? Any questions at this point?
No, sir.
Okay. Okay. So now, as I said, there are 3 pathways, and the 3 pathways, the initiation of complement is different for the 3 pathways, right? So complement activation, if we talk about the classical pathway, so this begins with the formation of antigen antibody complex. Okay, so for the classical pathway to start, some antigen or some antibody has to bind to the antigen. So when this antigen antibody complex is formed, then this classical pathway gets initiated. Okay. Next is your alternative pathway. It is initiated by cell surface constituents that are foreign to the host. Okay, so our immune system can sense that on this particular cell surface, the constituents, they are foreign to the host, right? So the immune system can sense that. So when it senses that the cell surface is different from what the host is having, right? In that case, the alternative pathway is initiated. And students, alternative pathway is antibody independent. Okay, so alternative pathway does not require antibody for its initiation. Okay, because in this case it is recognizing the cell surface constituents. Okay, so if they are foreign, then the alternative pathway begins, otherwise nothing happens. Okay, so this is antibody independent. And the third pathway, the lectin pathway, again it is activated by the binding of MBL to mannose residues on glycoproteins or carbohydrate on the surface of microorganism. Okay, so again in this case we will understand this later, what is MBL, what is other things. I won't discuss right now because then you'll get confused. Okay, and that would be too much to handle at this point. So in this case, lectin pathway gets initiated by a different mechanism when the host protein binds to a bacteria. Okay, and then this complement system gets activated. So again, you do not require antibody. So even this mechanism is antibody independent. Okay, so only the classical pathway is dependent on antibody. Alternative pathway and lectin pathway, they are not dependent on antibody for its activation. Is it clear? Because this is very important to understand. So which of the pathway is antibody dependent? Students, which of the pathway is antibody dependent?
Alternative pathway and lectin pathway.
They are antibody independent. I am asking which is antibody dependent.
Classical
Classical.
pathway.
Yes. So only the classical pathway is antibody dependent, because for this pathway to get activated, there has to be a formation of antigen antibody complex. And you can see in alternative pathway and lectin pathway, you do not need antibody. Okay, so antibody is not required here, antibody is not required here, but antibody is required in case of classical pathway. Okay, clear? Okay, another very important thing, as I said, that these complement proteins, they are present in our system, but they are not in their active form. Okay, so most components of the complement system are proenzymes or zymogens, which are inactive until proteolytically cleaved. Okay, so most of these complement proteins, they are present as proenzymes or zymogens. Okay, in their inactive form. Okay, and they will become active only when they are cleaved, right? So why this happens? Because I'm sure you have read enzymes in biochemistry somewhere. Have you read enzymes?
Yes.
Students, have you read enzymes?
Yes, sir.
Yes, sir.
Have you heard this term proenzymes or zymogens earlier?
No, no, sir.
No, sir. No,
Okay.
sir.
Do you know that in enzymes there is one active site?
Yes, sir. Yes, sir.
Okay. Okay. So I'm trying to find something just to explain you. Let me see. Okay, I found it. Okay. Now see, just suppose this is your enzyme. Okay. Now this is in its inactive form. Okay. Why? Because the active site is hidden. Okay. So what happens when it is proteolytically cleaved? Okay. Now on proteolytically cleaving this enzyme, the active site, this point, okay, this is exposed. Okay, and you can see what happened here. Two fragments are formed. One is the bigger fragment, another is the smaller fragment. Right? Clear? This is a proenzyme in its inactive form because the active site is hidden. Okay, and because of proteolytic cleavage, it will uh, get broken down into two fragments, right? The bigger fragment and the smaller fragment, and that leads to exposure of the active site. Okay, so what happens? Most components are proenzymes or zymogens, which are inactive until proteolytically cleaved, and it removes an inhibitory fragment that exposes the active site. Okay, so it removes the inhibitory fragment that exposes the active site. Okay, so now this complement protein is in its active form. Okay, clear?
Yes, sir.
Okay. Now, if you see the last line in this slide, you will see that mostly the smaller fragment is termed as A, right? So this smaller fragment is your A, okay, and the bigger fragment is your B, okay. So I think you can just remember bigger B, smaller A. Okay, so these complement proteins, as I said, there are a lot of different complement proteins. So इनके बहुत तरीके से namings हैं. So like the complement protein 1, complement protein 2, so we just say them as C1, C2, C3, C4. Okay, so in some other cases they are called as some factor, maybe factor B, factor D, right? And by sometime by trivial names like HRF, like the homologous restriction factor. So you will see that there are different ways the complement proteins are named. Okay. So peptide fragment formed by activation of component are denoted by smaller letters. So as I said, the fragments which are formed, so in this case, two fragments are formed, right? So one of them is the smaller fragment, which is A, and the bigger one is your B. Okay. Now let me just explain you this. Now suppose there is a complement protein, let's say for example C3, or let's say for example C4. Okay. Now, when the complement system gets activated, so just see here, they were in the proenzyme phase. So this is your zymogen in active form. And when it is proteolytically cleaved, it forms two fragments. Right? Okay, and as it is mentioned here, the smaller fragment is your C4A, and another one is your C4B. Clear? So it becomes C4A and C4B. Clear?
Yes, sir.
Okay. So as I said, if you take the example of C3, when it is cleaved, it becomes C3A and C3B. So students, in this, which is the bigger fragment?
C3B.
B.
So this is the bigger fragment and this is the smaller fragment, right? But C2 is exception. What does that mean? So let me make C2. So this will be broken down into C2A and C2B. So students, in this case, which is the bigger fragment? Students, which is the bigger fragment here?
C2A.
Yes. So just in case of C2, C2A is the bigger fragment. Okay. So it's an exception. It does not follow the rule which others are following. Okay. So otherwise for C3, C4, C5, you will see that C3B or whichever is having the B fragment is the bigger one with the exception of C2. Okay. Now the larger fragments, they bind target near site of activation, right? Whereas smaller fragment, it diffuses from the site and initiate localized inflammatory response by binding to specific receptors, right? So you can understand, just compare two individuals, one is very fat, okay, another one is very thin and small. Okay, now what happens? Obviously the fat one will not be able to move too much. Okay, so and the smaller one and the thin one, it can run fast and it can move to different place in a short period of time, right? So in this case also, in the complement system also, the larger fragments, they just bind to the target near the site of activation. जहां वो activate हुआ, उसके पास में जो भी cells होंगे, pathogen होगा, उस पे जाके activate होगा, right? Whereas the smaller fragment, right? So just imagine this. So smaller fragment, it will move away and do its work at some other place, and this will just bind to a pathogen nearby, okay, and get activated, right? So larger fragment, they bind target near the site of activation, whereas smaller fragment, they diffuse from the site and initiate localized inflammatory response at that particular point, okay? And complement fragment interact with one another to form the functional complex. Okay, so कॉम्प्लीमेंट के जो प्रोटीन्स हैं वो एक के बाद एक सब इंटरैक्ट करते हैं एंड देन दे फॉर्म द फंक्शनल कॉम्प्लेक्स. ठीक है, तो इसका जो फंक्शनल कॉम्प्लेक्स है उसके बारे में हम बाद में डिटेल में बात करेंगे. व्हाट डू यू अंडरस्टैंड बाय दिस फंक्शनल कॉम्प्लेक्स? उस फंक्शनल कॉम्प्लेक्स का फंक्शन क्या है, ओके, और उस फंक्शनल कॉम्प्लेक्स तक हम कैसे पहुंचते हैं, वी विल टॉक अबाउट दैट. Complexes having enzymatic activity are designated by bar over the number or signal. Okay, so इसका क्या मतलब है? Just for example, suppose if you have C4, so this becomes C4A plus C4B. Okay. Now the complex which is having enzymatic activity, right? So they are designated with a bar. So we put a bar over them. Okay. So if you see a bar over a particular complement protein, it means that it is enzymatically active. Okay. Clear? Clear till this point? Any question?
No, sir.
Okay, students. So if you have any question, you can just ask now, because the next class I will start with the classical pathway then. Okay, so you have to understand everything till this point so that it becomes easier for you to understand हम आगे जो भी discuss करेंगे. Okay, so I think in the meantime we can have attendance. Okay, so I will start with okay, Adish.
Present, sir.
Atoiba. Ayushi.
Present, sir.
Sam.
Present, sir.
Sam, you are not visible. Okay, Devanshi.
Sir, she is having network issues.
Okay, Ishita.
Present, sir.
Ishita, okay. Janvi.
Present, sir.
Kshitij.
Present, sir.
Manaswini.
Present, sir.
Okay, Devanshi. I've seen you. Namrita.
Present, sir.
Nandini.
Sir.
Nehal.
Present, sir.
Rupal.
Present, sir.
Okay, Sahiba.
Present, sir.
Sanjana. Okay, Shakti.
Present, sir.
Shivani.
Present, sir.
Tanvi.
Present, sir.
Tridiv.
Present, sir.
Vivek.
Present, sir.
Okay, Yukta. Okay, and Yuvraj.
Present, sir.
Okay. Okay, fine. So students, I'll just end the class now, okay? And we will meet after 5-10 minutes, and we can continue with complement system, or if you want, we can take some other aspect of the practical as well, okay? So it's up to you. We can discuss after 4-5 minutes, okay? So I'm ending the class now.